As we discussed in the previous unit, the process of transcription begins with this coding strand of DNA here at the top. The coding strand of DNA runs 5' to 3', and you can tell it is DNA because there is no uracil present. Complementary to the coding strand of DNA is the template strand of DNA that runs 3' to 5'. From our template strand of DNA, our wild-type mRNA can be created with the following reading frame. The mutant mRNA can be disregarded at this time. But the wild-type mRNA codes for the following amino acids: methionine, tryptophan, asparagine, alanine, and cysteine. Given the following DNA coding sequence, this is the fully functioning polypeptide protein that is formed as a result. However, when a single nucleic acid point mutation occurs in the mRNA, the chemical makeup of our protein changes, yielding potentially catastrophic defects to our protein. A missense mutation is a single point nucleic acid change in the mRNA that codes for a different amino acid in the protein sequence. In our wild-type mRNA, the third frame reads AAC. Our mutant mRNA undergoes an A to G transition to yield GAC in the third frame. And instead of AAC coding for asparagine, we have GAC coding for aspartate. A nonsense mutation is a single point nucleic acid change in the mRNA that alters an amino acid codon into a stop codon. In our wild-type mRNA, the fifth frame reads UGC. In our mutant mRNA, the fifth frame reads UGA. We have a C to A transversion, and instead of coding for cysteine, we have UGA, which is a premature stop codon, shortening the protein sequence from 5 amino acids to 4. A silent mutation is a point nucleic acid change in the mRNA that still codes for the same amino acid. In our wild-type mRNA, the fourth frame reads GCU. In our mutant mRNA, the fourth frame reads GCA. This is a U to A transversion, and as seen, GCU and GCA both code for alanine. A readthrough mutation is a point nucleic acid change in the mRNA that changes a stop codon to an amino acid codon. In our wild-type mRNA, the sixth frame reads UGA. In our mutant mRNA, the sixth frame reads UGG. This is an A to G transition, and so in changing this removes the stop codon and codes for UGG, which is tryptophan, CCC, which is proline, and the chain continues to elongate due to the stop codon being removed.